We're here on Vancouver Island near Campbell River, British Columbia. We're at the site of the STEMS silvicultural experiment. This is Dr. Sue Grayson, soil microbiologist at University of British Columbia. Dr. Grayson, can you explain a little bit more about this treatment that we see behind us?
Yes, well, what we have here is the dispersed retention treatment of STEMS 3. So this site was harvested in 2008, and this particular treatment is looking at dispersed retention where the trees are left roughly 18 meters apart for helping in the regeneration of the future seedlings on the site. In the background, over the other side of the lake, you can see the clear-cut treatment within this silvicultural treatments experiment.
What are the role of soil microorganisms in a forest ecosystem such as this?
Well, they're really vital components of the forest. We can't live without the soil organisms, and there's a immense diversity of organisms that we just know very little about in the soil, but we know they are vital for recycling of nutrients in the soil, for enabling the trees to grow, and they also produce and consume gases involved in global climate. So they really are a vital component, but we just know very little about them and the effect of harvesting methods on these organisms.
What are some of the challenges in terms of studying soil microorganisms in an environment like this?
Well, besides sampling in an area like this, just studying these organisms, because you can't see them with the naked eye. So a lot of times they've been ignored because you can't see them apart from the large macrofauna. But until sort of 15-20 years ago, we just didn't have any techniques to look at them. We could try and culture them on agar plates, but we now know that, you know, we've only been able to culture 1% of what's down there. There's billions of organisms in just a teaspoon of soil, so more organisms in that teaspoon of soil than there are people on Earth. And we only really know something about 1 to 10% of them.
One of the key ecological questions that investigators are trying to unravel is the relationship between biodiversity and ecosystem functioning. Can you talk a little bit about the relationship between soil microorganisms and their diversity and their effects on ecosystem functioning?
Yeah, well, we know, as I mentioned, so very little that in some cases we know there's a direct relationship between diversity and function. For example, with the nitrifiers, we also know which they oxidize ammonium to nitrate. We know something about another keystone group of organisms, the white rot or the wood rotting fungi, but as for other groups of organisms, we know very little about them or what they're doing and what are the consequences if we lose them, for example, with our management treatments. So one thing when we're looking at the dispersed retention treatment is that a lot of these soil organisms require carbon for growth, and they're getting carbon from these living trees, because from a living tree you have more readily available carbon coming from the litter materials through root exudation below ground, whereas currently in the Forest Practices Code all that's recommended to retain soil health after harvest is leaving slash on site. And we know that, you know, a lot of organisms, it's, you know, like eating tough cardboard, whereas, you know, with exudates it's much more juicy fruit material that is preferred particularly by bacteria. So there's certainly beneficial effects above ground of leaving trees, like for biodiversity for mammals, but also below ground, and nobody's actually looked at that before.